My overview of my research proposal on migraines and adeno-associated virus genetic intervention. So what are migraines? They're chronic headaches that typically last several hours to several days. It can depend on the severity and the person. They can be accompanied by symptoms like nausea, sensitivity to light and sound, and in severe cases vomiting. They're typically unilaterally located, but they can be bilaterally located, and typically sufferers describe an extreme throbbing or pulsing sensation in their head, like an extreme headache. And overall, this impacts their workplace efficiency and quality of life, so this can be a detriment to their familial relationships, their occupational satisfaction, their educational achievement, and many other factors that result from their anxiety and fear of another onset of migraines. So the previous methods for treatments include calcitonin gene-related peptide receptor inhibitors, which are basically, they're in clinical trials right now, but overall they act as an inhibitor for the calcitonin gene-related peptide that increases inflammation of the trigeminal nerve, which is implicated in the onset of migraines. And then we have the Tianusu capsules that are popular Chinese herbal medicine, and they have proven to be effective, but they result in adverse reactions and therefore poor adherence. There's also the administration of ketamine, which is basically a treatment that is a 5-HTIF inhibitor, or that's lasmiditan, sorry, and that's a highly selective receptor agonist of 5-HTIF. And then the ketamine administration basically reduced inflammation of the trigeminal nerve again. So my research question had to do with developing a clear understanding of the role that serum calcium levels play in the CNS, and basically there was a genome-wide association study that showed a moderate correlation between increases in serum calcium levels and increases in incidence of migraines in patients. So I use this to kind of base my hypothesis off of finding a genetic-based intervention that would be less dangerous than the previous interventions and more effective over the long term. And this would use adeno-associated virus and specifically engineered viral vectors targeted to modify the expression of the CASR gene, which is it regulates the expression, or the detection, it detects the calcium levels in the blood. So this would also investigate the role of the trigeminal inflammation in causing migraines, because this is found in a lot of studies to be the central cause of migraine onset. So I would do this with electrophysiological recording of the trigeminal system in mice models that have mutations in their voltage-gated calcium channels, and that would be to measure the effects of adenovirus administration. And to design the adeno-associated virus capsid, you would use cryo-electron microscopy, and to design the inside, the delivery materials that are in the capsid, you would use cellular assays and site-specific mutagenesis to design the specific viral vector that would be regulating the protein CASR. And the adeno-associated virus would be administered intravenously to the lab mice. And this would basically be used to see if the group that was administered the adeno-associated virus would have decreased activation of the trigeminal system, because the CASR gene is detecting more calcium and therefore inhibiting the release of more. And the ethical considerations for this experiment are the use of surgery on mice. Obviously, if it's a mouse model, electrodes will have to be inserted into the trigeminal nuclei to measure the activity of the trigeminal nerve. And during surgery, the proper handling of mice to minimize stress, use of anesthesia, and overall just to make sure the mice experience as little stress as possible. And housing mice is another consideration to be had to make sure this experiment runs ethically, because they are animals and a lot of stress comes from their environment. Thank you for listening.